Muchas gracias. Eran las palabras del presidente de Colombia, Iván Duque, a propósito de la temática que hoy vamos a abordar y que será el tema central de cada una de las conferencias, la lucha contra el Fusarium. Y a propósito de ello, hablando de Fusarium, nuestra siguiente conferencia virtual estará a cargo de Gert Kema, profesor de Fitopatología tropical. Su temática, Fusarium R4T, Amenaza Global para la producción de banano, desafíos y soluciones. Gert, bienvenido.
Okay, buenos días. Good morning, everybody. I hope you can hear me. Can you confirm? Yeah, okay, good. Well, good morning, everybody. Thank you very much for your kind invitation for me to speak at the 18th Convention of the International Conference of Banana, organized by IABE. So it's a pleasure to talk about our ongoing work on Fusarium Wilt, TR4, and I was asked to talk about the risk for the global banana industry, the challenges and the solutions. So once more, thank you very much for the invitation, and I will move on from here. So first of all, I think it's good that we set a scene. So what you see here, and I hope you see my pointer, is a Cavendish plant that is affected by Fusarium wilt caused by tropical race 4. So the very typical symptoms, sclerosis of the leaves that eventually will drop down. And in this process, the plant is completely colonized by spores of Fusarium fungi that are spreading once the plant is being harvested, but it's also producing spores in the soil. And what is important to keep in mind, it's a typical soil-borne disease, so it travels slowly, except of course when vectors are involved. And what we know from the global epidemiology is that people are the main vectors, and that's why you have also massive international or even intercontinental germs of the fungus. So by producing the resting spores, it is very persistent, but TR4, for instance, is an absolute killer in banana, but can also infect non-host plants like weeds in plantations that do not show any symptoms, and the fungus is not killing those weeds. So it is very versatile in maintaining the inoculum of the fungus in the plantation or in the soil. So it's a true survivor. What is also important to keep in mind is that the cause of the Fusarium wilt epidemic in Gros Michel was really caused by a suite of different Fusarium genotypes, and I will show you the details later on. Whereas Fusarium wilt in Cavendish is caused by a single clone, and we call that clone usually tropical race 4. So that is just the facts to start off. Now let's go to the risk. The risk is this. We just have to go back into history in the 50s. If you Google on Panama disease of Fusarium wilt, you will see that Gros Michel was wiped out by this suite of race 1 strains in the previous century. And what you see here is a photograph that was shared with me by Professor Randy Plutz of the University of Florida in the US, where they controlled Fusarium wilt by inundation. Inundation generates an anaerobic condition in the soil, so there is no air, so everything suffocates, including fungi or any other organisms in the soil that need oxygen, so the aerobic organisms. And then later on, these fields were again planted to Gros Michel. This is exactly the same field, but in those days there was not yet tissue culture, so those plants were multiplied by suckers. Usually, or many of these suckers were already infected by Fusarium, but you just didn't see the symptoms yet. And that really boomed the epidemic. We all know what eventually happened. Fusarium wilt wiped out Gros Michel across Central America, really truly causing havoc, social insecurity, land claims and everything that came with it. And eventually, fortunately, Cavendish was discovered. Cavendish was not bred by a company or by an institution. Cavendish bananas are wild clones that were collected by the French in Southeast Asia, most likely in Vietnam, only maintained and survived in botanical gardens, and eventually ended up in the botanical garden of Chatsworth House in the United Kingdom, in Derbyshire, where it was actually grown for the guests that were visiting the Duke in Derbyshire at Chatsworth House to serve them banana fruits. And only later on it was found out that Cavendish bananas were super resistant to the strains that caused Fusarium wilt in Gros Michel. And since then, Cavendish gradually emerged and was embraced also by the industry as the solution for Fusarium wilt. Up to the level, as you can see here now in this small pie chart, right now more than 50% of the global production is dominated by Cavendish, and more than 95% is it takes 95% of the global export business. Well, here you see an example. This is a massive global monoculture. This is an example from Urabá in Colombia. I'm sure we have very similar pictures from Costa Rica and also from Ecuador as the largest exporter in the world. And we all feel, of course, having this massive monoculture is an immense risk. We also all know that Cavendish is very susceptible to black sigatoka, which results in increasing number of cycles. But on top of that, tropical race 4 now is a true killer of Cavendish. And that is not very recent. That is not very recent. This was already seen in the 1960s in Taiwan, but was really considered by the industry a faraway story until it really started disseminating into remoter areas, so outside Southeast Asia. And I come back to that. But it's very important to underscore that Cavendish is still very resistant to the strains that caused havoc in Gros Michel. And that is exceptional for a cultivar to be resistant to a fungal strain or fungal strains for over 70 years. And it's very important also to underscore that Tropical Race 4 is not a mutant of those strains. Tropical Race 4 is truly a new strain that emerged in Southeast Asia and is conquering the world. And so the solution of Cavendish to the Race 1 strains is phenomenal and still holds. And so it's very understandable that this was really embraced eventually by the industry and that all the problems of Fusarium wilt were over in general, because you could grow and still can grow Cavendish on the most infested soils with Race 1 strains without any problem. So it was a fantastic demonstration of the power of natural resistance, which is environmentally sound and apparently in this case also durable. But this happened later on, as I said. TR4 showed up in the 60s already in Taiwan, since then disseminated across Southeast Asia, and from about 2012 onwards, also in the Philippines. So the banana belt of Cavendish production in Mindanao in the Philippines is heavily struck by TR4. And here you see an example. This is a Cavendish plantation that is devastated by Tropical Race 4. If you cut an infected plant, this is the cross-section that you will see. So all those yellow parts that you see here, this is all colonized xylem vascular strands. Here as well, all these darker spots, this is heavily colonized by TR4. And eventually, as you can imagine, this completely occludes the transport of water and nutrients. The plant starts wilting, has produced billions of spores in the stem, and eventually dies. So it's a truly vascular disease. Well, as I said, this is then the summary story, and that's the risk. In the 60s was already seen in Taiwan. Since then, it has spread across Southeast Asia, We reported it the first time outside Southeast Asia in 2014. Meanwhile, we had developed molecular diagnostics, so it didn't take months to determine whether a strain is TR4, but we could simply diagnose it in a day by DNA isolation and running PCR diagnostics, very similar to what we currently do for the coronavirus globally. We use exactly the same tools. All those green dots are cases where we have confirmed using our diagnostics that TR4 is on the move. And of course, the big shock was when it entered in Colombia in 2019 and more recently in Piura in Peru. So yeah, this is the ongoing story. So we are dealing with a true pandemic. It's a soil-borne disease. It moves slowly, but all those international jumps and intercontinental jumps also here in Africa, which really threats the banana production in East Africa here, where banana is a staple food. So not just a fruit, but very important for the daily intake for millions of people. These are truly jumps that are where humans are the vectors. So either by contaminated tools, footwear, and maybe sometimes even transportation of plants, although that is, I think, a very, very minor share. So to summarize, the Fusarium wilt epidemic that we currently experience is a true repetition of history. It started with rain 1 strains in Central America. Now it's TR4. And the example I gave in the Philippines, but as you can see here, TR4 is spreading. And of course, the risk is that it will continue spreading in Central American countries. So we really have to focus very strongly on quarantining areas where it has been found, prevention that it will come into other areas, and we need tools for it to really be able to do that. We have to conclude from this picture that so far we have been very unsuccessful in quarantining TR4, because despite everything that's being done, it has spread, even into an exhibition in the UK and England. As we all know, that's not a banana producing country. Nevertheless, it entered also in that particular location. And so this really underscores that it is extremely difficult to prevent the entry of TR4. Now, what are the challenges? So that's the second subject of my talk. Well, the first challenge really is that it continues disseminating. And let me just give you this example. Here we are watching now the Mekong Delta in Southeast Asia. So this is the Yunnan province in China, bordered by Myanmar, Laos, and Vietnam. Yunnan is heavily struck by TR4. So the banana producers that can afford it start new plantations in Myanmar, Laos, and Vietnam. In all those plantations, TR4 was diagnosed. So we sampled Yunnan and plantations here. And as you can see, if you sample those isolates, you isolate DNA and you sequence it, you see that these isolates are very, very similar. So the strain from Myanmar, Laos, Vietnam is very similar to the strain from China, but also to the strains that we identified in Lebanon and Jordan, in the Philippines, etc. So this really underscores we are dealing with a single clone that is spreading. And if you then look at in more detail, you really see that the strain from China really sits in the same corner of this principal component analysis with the strains of Laos, Vietnam, and Myanmar. Similar here for Pakistan and the Philippines, and the strains from Lebanon and Jordan. And this really underscores that this is human contamination. So growers, Chinese growers expanding into these countries, bring their own personnel, their own equipment, are ignorant about the problem of TR4 or don't take sufficient precautions and spread the disease to those other countries. And this is an immense risk. And one of the problems, of course, is that plants can be infected before you see the symptom, but also then the soil is already contaminated. I have walked in plantations in the Philippines where I extracted DNA from the soil under my shoes where we directly diagnosed TR4. So by just walking in plantations and moving from one country to the other, you of course spread the disease. Now, that's of course also the risk, an immense risk currently ongoing in Latin America and Colombia and in Peru. And I haven't even not yet talked about irrigation water. Water is a super efficient method to disseminate the disease. We know from floods after typhoons in Mindanao, for instance, Typhoon Pablo that occurred a few years ago, when massive areas were flooded, you really see the front of the disease that really coincides with the front of the floods. So water is an excellent way to disseminate the disease, and that's why we have to be so careful also with irrigation. Now, this is the current situation, or this is a map of the Piura area in Peru. So the entire valley is about 30,000 hectares. There's about 8,000 hectares of banana. And this is really considered as one big farm because it's all fed by this water dam here that really irrigates the entire banana producing area over there. And you can imagine once TR4 shows up there, there's an immense risk to all those farmers. And unfortunately, TR4 has shown up in the Piura area. So what we have done in collaboration with Opus Insights, it's a Dutch company and a couple of other partners, and Wageningen is also participating, is to map the entire area by aerial mapping, flying planes, not drones, because the throughput is way too low. And satellites, you always have the problem that you have a reduced resolution, at least until now. And of course, many times there is overcast, so you cannot look through the clouds. So we fly planes at max 1,000 meters. You see here the resolution. So if we zoom in here on this area, we see here a couple of suspicious spots, so empty spots. And of course, then from the air, you can wonder, hey, what's going on? You zoom in, and then you see this spot here. There's already some canals being produced. So you really wonder what's going on in that location. So this would be a case where you would inform, for instance, SENASA and say, hey, here we have a suspicious spot. Can you go there, sample, and see whether this is caused by TR4? Same story here. This spot here. Also here you see kind of canals being produced. And as you can see, the resolution is pretty good. So you even would be able to identify plants that are chlorotic. So the throughput here is that you essentially can map thousands of hectares in a day by just using multiple planes. So this is a very powerful tool for risk management. Here's another example. So these are individual growers, individual farms of about 1 hectare each. So this one looks pretty good, but this one looks worse. And so here also you could really wonder, would it be worthwhile to send personnel over there or government officials, because they are responsible for diagnosing TR4 and also for the quarantine actions that should follow. So they could go here, watch these plants, sample them, and see whether these are affected by TR4. So in our view, aerial mapping is extremely powerful and useful, particularly now, because the threat is at your doorstep. So you really would like to map out risk zones and see what is the watershed in this area and which regions are potentially at risk. And of course, you need diagnostics. So fortunately, there are plenty of diagnostics now developed. This was the first one that we developed and published in 2010, and which has been commercialized. And having a commercial diagnostic is extremely important because everybody uses exactly the same tools and methods. So you can compare data. But of course, you need multiple diagnostics. So this was the first one produced in 2010. 2019, we published a LAMP diagnostic, which doesn't have a high throughput, but you can take it in the field, and it gives you an output within 15 minutes. And eventually, we would like to have this diagnostic. So as you can see, this is exactly the same technology that's used for the coronavirus. So this is high throughput, high numbers. This is low throughput, but fast. And this is very fast. So you can take it in your pocket and test it yourself in the field. So once you diagnose TR4, of course, you have to inform officials, and then you have to start the whole quarantining. Now, multiple diagnostics is very important because one diagnostic should always confirm the other. So the one that we published in 2010, what you see here actually is a cartoon of the genome of each chromosome of TR4. So this is chromosome 1, chromosome 2, etc., to chromosome 12. And the red bullets are what we call the telomeres, the beginning and the end points of the chromosomes. So the first diagnostic had its target here on chromosome 4. But the new one that we developed has its target on chromosome 11. So what it really means is if this diagnostics is positive, this one should be positive as well. And so this really reduces the chance for false positives or negatives. So increases very significantly the accuracy of molecular diagnostics. And of course, this is important because once TR4 shows up, you have to really screen thousands of samples to make sure that you have an appropriate quarantining strategy. Okay, the second challenge is we have to continue research and to publish our data, share as much as we can. So we started what we call a research topic at the Frontiers in Plant Science. I looked it up yesterday, more than 200,000 views went through this research topic. So it's really a resource for many people to see what is now the latest in a particular area. So there are 13 articles there by multiple authors. And I think this was a great initiative that we embraced once it was brought to our attention. And this is very important, particularly right now. And so again, in relation to the Corona strategy, there was an article in Nature Briefing last year in April where it said, you know, first we must stop legitimizing pseudoscience. And I have to say also with regard to TR4 and banana, there's a lot of pseudoscience being disseminated. And the only way to deal with that is to publish accurate data and really swamp the pseudoscience by good research. Now, I have worked for many years in wheat, and I would like to share this comparison with you. If you look into the genetics of wheat, more than 8,500 genes have been described and validated, and more than 1,000 of these genes are involved in diseases and pests. If you look to banana, such an important food crop, there are no more than 25 genes really described or validated, and only 2 are involved in disease resistance. And they deal with Fusarian wilt. There's not a single resistance gene identified and validated for black sigatoka, for instance. So where we are in banana research is no comparison to many other food crops. So that really means we need lots of people to perform and to train people to do appropriate research. Now, if you would read this book by John Saluri, which is on the first Fusarian wilt epidemic in Central America, you will see that many of the things that are currently being done were also tried in the previous century, and eventually everything failed except Cavendish. Cavendish was resistant. And that was the major reason why the banana production in Central America was saved. And that's why we also have to work towards new varieties with resistance to fungicide, to the fungal diseases we talk about. Now, what we have done is explore what is now the diversity of Fusarium around the world. And so in an attempt to really have a global map of diversity, and how can we use that to build new varieties that are resistant. So one of my PhD students, Nani Mariani, she originates from Indonesia. She sampled Fusarium across Indonesia. More than 200 isolates, 50% of those strains is TR4. So TR4 is disseminated across the archipelago of Indonesia. But what she also showed is that we have multiple species. So this was previously all called Fusarium oxysporum specialis cubense. But the latest insights is that this is actually not right. Actually, the race 1 strains are really individual different species. And I will show you that in the next slide. And here you see TR4. That's again a different species, really significantly different from the race 1 strains. So we also looked globally. This was done by Fernando Garcia from Colombia and Nadia Ordóñez from Ecuador. She's from just a little bit south of Guayaquil. So altogether, we had about a collection of 700 isolates, and all these isolates have been genotyped. And what I mean with genotyped is that we use DART-seq, which is a high throughput genotyping by sequencing, but many of those isolates were also completely sequenced. And that is now here represented in this map. So this is a diversity phylogram, as we call it. Each line here, each bullet is a different strain. I'm not going to the details, but what is important to show is this. All the strains that are under this part is all race 1 strains. And as you can see, you know, this phylogram really shows that this group is different from this group, is different from this group, from this one, etc. So here you see the genetic diversity of race 1 strains, whereas this is TR4. So here we have about 150 isolates of TR4, genotype and sequence, and as you can see, they are very, very similar. You don't see these different clusters. This is just one cluster, which really underscores TR4 is a single new species. The race 1 strains is multiple species, and that is of fundamental importance. We concluded that Fusarium dissemination across the world has most likely taken centuries and all originates here from the center of origin of banana. So here is where you find the largest diversity also for the fungal pathogens of banana, including Fusarium, that was most likely disseminated on all the different continents by the transport of infected plants. And that's actually still ongoing, right? So we transport plants. This was done by humans. They take the diseases along. Now we fly around the world. We don't take plants any longer, but if you don't take care and you work with contaminated shoes, footwear, tools, etc., you disseminate the disease. Okay, to continue, what we need to look for is diversity in the hosts. As we said, Cavendish is a global monoculture, extremely risky. But if you look, for instance, in the island of Sumatra in Indonesia, all the orange dots here are locations where wild bananas have been explored by another PhD student, Fajar Ahmad. And these wild banana species, this is all Musa acuminata, so one of the ancestors of Cavendish. So all these bananas have been described and also genotyped, and eventually this is a rich resource of diversity. Now, what Fajar did, he looked into one of those ancestors, Musa acuminata malacensis, and here you see the situation as we know it. Gros Michel, very susceptible to race 1 and to TR4. This is race 1 and this is TR4. Cavendish, super resistant to race 1, very susceptible to TR4. Yet, the difference in phenotype, nobody knew which genes or how many genes are involved in this extreme level of resistance. So this is what Fajar addressed. He made crosses, we analyzed the populations, published it, and what we have seen is that on chromosome 10 of banana, there is a gene that is responsible for this high level of resistance. So this is the second gene that was described for resistance to Fusarium. Of course, now we are in the process of cloning the gene because this gene is indispensable in any new variety. You need resistance to race 1, period. You cannot do without it. On top of that, you need resistance for TR4. The second gene is the gene described by James Dale in his group, and we were involved as well. This is also a gene from Musa acuminata malacensis, which gives a very high level of resistance to TR4. So this is the wild type. Williams tested with TR4. He cloned the gene from Musa acuminata, put it in Cavendish, and here you see the result. So this is a genetically modified Cavendish, highly resistant to TR4. The question, of course, is this the way forward? In my opinion, this is a proof of concept, but not the way forward, because sustainability is not secured by replacing Cavendish 1 by Cavendish 2. For instance, Cavendish 2 is still very susceptible to black sigatoka. So the only way to simultaneously address disease problems is plant breeding. There you look at problems simultaneously And of course, what we shouldn't forget, the underlying problem of these disease problems, and that's why we work now towards the solutions, is genetic uniformity. Cavendish all over the world is extremely risky. If you look at any other crop, that hardly ever occurs, maybe only in forestry. But plant breeders are continuously breeding new innovative varieties to replace the older ones. There is not a single crop that is there for 70 years. And so sustainability comes with diversity. We have to replace Cavendish by a panel of new, diverse, competitive, resistant varieties. And the basis for that is Southeast Asia. There you have all the diversity of banana, but this is completely untapped. This is not used in breeding right now. Not because it's impossible, but because we have too few hands. So we need more breeding efforts. There's only a handful of breeding programs around the world, whereas in other crops there are hundreds of breeding programs. So this is where our focus is, sustainability on the long run. So we have to stop with a monoculture and change that into a sustainable, diversified banana production system. That's why we have started the Breeding Initiative, a company with Chiquita Kichin and a startup in Wageningen, Musáceas. And the name of the company is Yellowway. So Yellowway is a true breeding company using the latest genetic tools to speed up breeding. So that doesn't mean we produce genetically modified bananas. No, we use molecular and hyper-modern tools to speed up classical breeding. And that is urgently required. One other way, and I'm almost finished with my presentation, is of course to see other ways of innovating banana culture. I recently talked to a tomato breeder and he said, you know, the tomato yields since the last 25 years have quadrupled. But 25 years ago, we grew tomatoes in a flat greenhouse, and now we grow them in very tall greenhouses on rock wool substrate. And so if you look at the overall production gains in tomato production, maybe 50% is due to genetics and plant breeding. The other 50% is due to innovating the culture of tomatoes. So this is what we have taken on, and we have started growing bananas on substrate. And if you grow banana on substrate, you really lift the crop from many problems. You lift it from nematodes, you lift it from moco, you lift it from Fusarium, you give exactly enough water, you give exactly enough nutrients, you don't have weeds. So this could be a true potential innovation. We have explored it, and this is what you see here in this movie. So just watch the movie for 1 or 2 minutes. Now, this movie was a trial. What we have now done is what you see here. We have built a greenhouse very close to Wageningen, where we repeat the experiment, but we grow Gros Michel, we grow Cavendish, we grow a plantain, and we grow cooking bananas. No diseases, no Sigatoka. Fantastic plants with big bunches. November 24, we are going to harvest those plants. And I'm sure many bananeros among the audience will say, you're crazy, we're never going to grow bananas on substrate. But I'm sure that was also told to tomato growers 25 years ago. I'm not saying that we have to swap altogether to growing bananas on substrate, but if it's only a few forerunners, a few niche markets where we can try this. So that's why we have now started planning field experiments in Guatemala and the Philippines to demonstrate the power of growing bananas on substrate. Of course, there we don't grow them in the greenhouse, we grow them in the field, but on substrate, and we irrigate them to collect data to see what is now the economic feasibility of growing bananas on substrate. Because I really believe we have to gain time. Because, you know, a breeding effort is nice, but it takes 10 years before you really can bring new varieties to the market. And we have to gain time. We have to make sure that you can continue banana production. So we have to first slow down the TO4 epidemic, prevent that it enters in any other new area. So we have to map it. We have to identify risk zones. We have to go down there and diagnose it. But also look into alternatives of cultivation. If we can slow down and prevent entry of TO4 by growing part of the banana belt on substrate, we are doing a great job. This is gaining time, and this will eventually give us the possibility to release new varieties that can compete with Cavendish in quality and primarily also in disease resistance. So with that, I'm ending my story. We're also writing books, so if you have any interest in, for instance, cultivation techniques, this was volume 1 of the book series. Volume 2 is on germplasm and genetic improvement, and we currently work on volume 3, which is about pests and diseases. So feel free to contact me if you're interested in these books, or go to the website of Burley and Dodds. With that, I would really like to thank you for your attention. I would really like to also acknowledge our partners and collaborators and funders of the research we have conducted in the Netherlands. And with that, I also would like to thank my team at Wageningen University, and I thank you very much for your attention. Thank you.